somewhere between 90 and 100%. That's the range geneticists have documented, tribe after tribe, when testing blood type frequency across indigenous populations of the Americas. Not a strong majority, not a noticeable trend. In some communities, tested repeatedly across decades of research, essentially everyone carries the same single blood type, O. To understand how strange that number actually is, you need to know what the rest of the planet looks like by comparison. Globally, type O sits at roughly the most common blood type, but nowhere close to universal. Closer to 45 or 50% of the world's population, mixed everywhere with A, B, and AB, in every country researchers have ever surveyed. There is no other continent-scale population on Earth that shows anything close to the near-total uniformity found across the Americas. And here's the detail that makes this genuinely strange, rather than simply interesting. Geneticists don't think type O is even the original ancestral version of human blood. They think it's a mutation, a broken copy of a gene that used to do something else and now does nothing at all. So the real question isn't why some indigenous communities carry type O blood. It's why an entire hemisphere ended up almost uniformly carrying what started out, in strict genetic terms, as an accident. To understand why that accident matters, you first need the basic biology of what a blood type actually is. Human blood carries tiny molecular markers called antigens sitting on the surface of every red blood cell, and the ABO system that defines your blood type comes down to just one gene on chromosome 9, with 3 possible versions. The A version builds an A antigen, the B version builds a B antigen, and the O version, according to decades of published research including the landmark molecular studies that first sequenced these genes in 1990, builds nothing at all because it carries a small deletion, a single missing letter in its genetic code, at a specific position researchers call 261 del G. That single deletion throws off the entire reading frame of the gene, causing the cell's protein-building machinery to stop early and produce a truncated, completely non-functional protein instead of a working antigen. In plain terms, the O allele isn't really its own distinct blood-building instruction. It's a broken copy of the A gene, one where the assembly instructions get cut off partway through, leaving the cell surface blank. That blankness is exactly why type O blood works as what doctors call the universal donor. With no A or B antigen sitting on the cell surface for an unfamiliar immune system to react to, O blood can generally be transfused into people of any other blood type without triggering the dangerous immune response that would occur otherwise, which is why hospital emergency rooms reach for type O blood, particularly O negative, when there's no time to test a patient before a transfusion. Genetic evidence strongly suggests the A allele, not O, is the ancestral version, since our closest living relatives, chimpanzees, also carry versions of A and O genes, while the specific deletion that defines the common human O allele appears to be unique to the human lineage, which means, roughly speaking, blood type O started out as a mutation, a broken instruction manual that somehow ended up becoming the single most common blood type on the planet, and, in parts of the Americas, functionally the only one. So why did the Americas end up so different from everywhere else? The most widely supported explanation, researchers have converged on traces back to how the Americas were first populated in the first place. And here, the story genuinely earns the word dramatic. The leading model for how humans reached the Americas involves small groups crossing from Siberia into Alaska, likely during a period called the Beringian Standstill, when falling sea levels during the last ice age exposed a vast land bridge, and early populations may have paused there, isolated, for 1,000 years or more, before finally moving south into the continent. A 2023 genetic analysis published in the Journal of Archaeological Science identified a specific O allele variant, distinguished from its Asian source population by a single mutation that appears to be essentially exclusive to Native American populations, and largely absent even from other populations like Arctic peoples, who share that same broader Beringian ancestry. That detail matters because it suggests this particular version of the O gene became fixed, meaning it spread to essentially the entire population, specifically during or shortly after that isolated standstill period, rather than simply being carried over unchanged from a diverse Asian source population. Here's where population genetics gives us a well-understood mechanism for exactly how something like that can happen, and it doesn't necessarily require any survival advantage at all. When a very small group of people finds an entirely new population, whatever genetic variants that small founding group happens to carry get passed forward and amplified across every subsequent generation, a phenomenon geneticists call the founder effect. If the specific individuals who crossed into the Americas and successfully reproduced happen to carry a disproportionate share of the O allele purely by chance, that skew could get locked in and amplified over thousands of years, especially once the Americas became functionally isolated behind two oceans, with essentially no new genetic material flowing in from outside populations to dilute or rebalance the mix. Combine a founder effect with genetic drift, the random fluctuation of allele frequencies that happens naturally in any population, especially a small one, generation after generation, and you have a scientifically well-supported, thoroughly tested explanation for why one blood type could end up dominating an entire hemisphere without needing any dramatic survival story attached to it at all. Now, here's where the story gets more contested than a simple, tidy narrative would suggest, and it's worth being honest about that rather than picking the most dramatic explanation and presenting it as settled fact. A separate and genuinely significant hypothesis holds that natural selection played a major additional role, not necessarily during the original migration, but much more recently, following European contact. Ancient DNA research, including a 2008 study published examining genetic material recovered directly from pre-contact skeletal remains in eastern North America, was specifically designed to test this question, comparing ABO gene frequencies in populations that lived before European arrival against the frequencies found in their modern descendants. If the near-universal type O frequency, seen in modern indigenous populations, had been produced primarily by the catastrophic population collapse that followed European contact, the devastating smallpox and other epidemics that killed an estimated majority of indigenous people across the Americas in the century following 1492, with survivors happening to skew disproportionately toward type O, you'd expect ancient pre-contact remains to show meaningfully more genetic diversity in blood type than their modern descendants do. Researchers who've examined this question directly note that current evidence doesn't yet fully separate these three competing explanations: an ancient founder effect during the original migration, genetic drift accumulating over thousands of subsequent years, or a more recent population bottleneck driven specifically by the post-contact disease epidemics, and probably some combination of all three contributed. What's genuinely fair to say, and what the field's own leading researchers are careful to state, is that this remains an open, actively studied question rather than a single settled dramatic answer, and the popular idea that ancient indigenous populations were somehow specifically selected across millennia to resist history's deadliest plagues overstates the certainty of what the genetic evidence actually shows. The honest version of this story is less about ancient warriors defeating disease through blood and more about small founding populations, isolated for thousands of years, whose genetic makeup then experienced one further brutal narrowing during one of the deadliest demographic catastrophes in recorded human history. That distinction matters, but it doesn't make the underlying story any less remarkable, because what modern genetics has confirmed with real confidence independent of exactly which mechanism dominated, is the sheer scale and consistency of the pattern itself. Blood type surveys conducted across dramatically different indigenous communities, from Arctic populations in northern Canada to groups living deep in the Amazon rainforest, separated by thousands of miles, wildly different climates, completely different diets and languages with no direct relation to each other, still show the same overwhelming skew toward type O again and again, independently, across study after study. That kind of consistency across such geographically and culturally distinct populations is exactly the signature you'd expect from a trait locked in near the very root of the population's family tree, at the original founding bottleneck, rather than something that developed independently and repeatedly for unrelated local reasons in each separate region. There's a genuinely interesting and separately documented layer to this story involving high-altitude populations specifically, and it's worth being careful here too about what's established versus what's still speculative. Indigenous communities in the Andes have lived at extreme elevation, often above 12,000 feet, for thousands of years, and their bodies show remarkable, well-documented physiological adaptations to that environment's dramatically reduced oxygen availability. Some researchers have proposed that blood type O might interact in some way with those altitude adaptations, potentially through subtle differences in how easily blood flows through narrow vessels. It's important to be clear that this specific hypothesis remains genuinely contested and isn't broadly accepted within mainstream hematology, and no consensus currently supports blood type itself as a meaningful driver of high-altitude adaptation, which is far better explained by well-documented genetic adaptations in entirely separate genes governing oxygen transport and red blood cell production. The overlap between high type O frequency and high-altitude populations in the Andes is real and documented. Whether one causes or influences the other in any meaningful way remains unproven. What is on considerably firmer scientific ground is a broader, well-established principle connecting ABO blood type to infectious disease susceptibility generally, even if it doesn't map cleanly onto the specific America story the way some popular retellings suggest. Multiple peer-reviewed studies have documented that certain pathogens, including strains of malaria, appear to interact differently with red blood cells depending on which ABO antigens are present, with some evidence suggesting type O carriers experience somewhat reduced severity in certain malarial infections, likely because the parasite's specific mechanism for anchoring to and rupturing red blood cells works less efficiently without the A or B antigen scaffolding present to exploit. Malaria wasn't a major pre-Columbian threat across most of the Americas the way it was across large parts of Africa and Asia, so this specific mechanism doesn't directly explain the pattern researchers see in indigenous American populations, but it does establish with real scientific backing that blood type genuinely can matter for infectious disease outcomes in at least some documented contexts, which is part of why the broader selection hypothesis for the Americas, while unproven in its specifics, isn't dismissed as implausible either. Modern genetic technology has done something else entirely alongside untangling these evolutionary questions. It's given indigenous communities and individuals genuinely powerful new tools for reconnecting with disrupted family and tribal histories, tools that would have sounded like science fiction only a generation ago. DNA analysis has confirmed direct genetic connections between living relatives and specific, historically documented ancestors, closing gaps that oral family history alone had never been able to fully verify. For many indigenous families, this kind of genetic confirmation carries weight well beyond simple curiosity. Colonization, forced relocation, and generations of disrupted or deliberately destroyed records severed countless people from documented tribal lineages, and DNA testing has become one of the only remaining tools capable of helping some families reconstruct connections history worked hard to erase. It's also worth being direct about the fact that this technology carries real practical and ethical weight attached to it. For many indigenous people, genetic ancestry evidence can directly affect access to formal tribal recognition, resources, and rights specifically tied to documented heritage, meaning a DNA test in these contexts isn't simply a curiosity purchase. At the same time, the same genetic data capable of reconnecting people to lost family history can also be collected, stored, and in some cases, commercially sold by private testing companies, and questions about consent, data ownership, and who ultimately controls that deeply personal information remain some of the most actively contested ethical issues in the entire field. Researchers and indigenous communities working directly together on how this data gets collected, stored, and used represents, according to many scientists in the field, the clearest path toward this technology becoming a genuine tool for reconnection rather than another form of extraction. It's also worth noting, with appropriate caution, that not every dramatic genetic claim circulating publicly about indigenous American ancestry carries the same scientific weight as peer-reviewed academic research. Commercial DNA testing companies have occasionally produced individual results suggesting unusually deep or unconventional ancestral timelines for specific customers, results that sometimes challenge the standard, well-supported migration models. It's important to be clear that findings released by commercial companies outside the normal academic peer review process, where other researchers can independently examine the methodology and data, carry considerably less scientific certainty than published peer-reviewed studies, and claims of this kind remain genuinely contested among archaeologists and geneticists rather than broadly validated. What is exceptionally well established and genuinely one of the more remarkable findings in the entire field of ancient DNA research over the past decade involves a discovery that has nothing directly to do with blood type, but everything to do with just how much farther and how much earlier human populations moved than anyone previously assumed. In July 2020, a team led by Alexander Ioannidis at Stanford, publishing in Nature, analyzed genome-wide data from more than 800 individuals across 17 Polynesian island populations and 15 Native American groups along the Pacific coast, specifically searching for genetic evidence of ancient contact between the two populations, a question that had been fiercely debated by archaeologists and geneticists for decades, ever since the Norwegian explorer Thor Heyerdahl first controversially proposed the idea in the mid-20th century. The genetic evidence the team found was unambiguous. Native American ancestry appears clearly within the genomes of individuals across multiple eastern Polynesian island populations, and by carefully analyzing the length of these shared ancient DNA segments, a technique that lets researchers estimate roughly how long ago two populations mixed, the team dated a single significant contact event to approximately 1200 CE, and traced the source of that Native American ancestry specifically to populations most closely related to the indigenous inhabitants of present-day Colombia. That means roughly 8 centuries ago, someone crossed thousands of miles of completely open Pacific Ocean, made contact with people who had never encountered them before, and left a genetic legacy still clearly detectable in living Polynesian populations today. The clues had actually been sitting in plain archaeological sight for a very long time before the genetics confirmed them. The sweet potato and bottle gourd, both plants native to the Americas, show up unmistakably in the Polynesian archaeological and linguistic record, with no other coherent explanation for how an American crop reached islands separated from the mainland by thousands of miles of open water. For years, that evidence remained hotly contested, dismissed by some researchers as possible coincidence or accidental ocean current dispersal rather than genuine human contact. The 2020 genetic study effectively settled that decades-long argument. So here's where the actual evidence lands, separated clearly from speculation. It's confirmed through molecular genetics research stretching back to the early 1990s that the O allele responsible for type O blood arose from a specific deletion mutation in an ancestral A-type gene, producing a non-functional antigen and, as a direct consequence, the universal donor property still used in hospitals today. It's confirmed through decades of independently repeated blood typing studies and more recent genomic analysis that indigenous populations across the Americas show blood type O frequencies far higher than anywhere else on Earth, in some documented communities approaching statistical uniformity. It's confirmed that a specific O allele variant appears essentially unique to Native American populations, consistent with it becoming fixed during the population's original isolated founding migration. What remains genuinely open and actively debated among researchers is the precise combination of forces responsible for that extreme uniformity, an ancient founder effect during the original crossing, thousands of years of subsequent genetic drift in an isolated hemisphere, a later and considerably more severe narrowing driven specifically by the devastating post-contact disease epidemics, or some blend of all three, with current scientific evidence not yet fully resolving which factor deserves the most weight. And entirely separate from the blood type question, but drawn from the very same broader field of ancient population genetics, it's now definitively confirmed that indigenous American and Polynesian populations made direct genetic contact roughly 800 years ago. Evidence of an oceanic voyage bridging 2 worlds long before anyone in Europe knew either of them existed. The next time someone tells you O is just the plain simple blood type, sitting quietly in the corner of the blood bank, you'll know that's not really the full story. It's a genetic record, imperfectly understood but genuinely remarkable, of an isolated migration across a vanished land bridge, thousands of years of a hemisphere cut off from the rest of the world. And, separately, proof that someone 8 centuries ago crossed an entire ocean nobody thought had been crossed at all. If you're type O, does knowing this history change how you think about something as ordinary as your own blood type? Let me know in the comments, and if this one surprised you, go ahead and like, subscribe, and turn on notifications so you don't miss what we uncover next.